Hey, hi everyone. Yeah, so I'm presenting today about a toolbox, or wouldn't really call it a toolbox, like a JavaScript tool to allow.
Oh, Alex, we lost your audio, or at least I did.
Hello?
Hello? Okay.
Cool. Yeah, sorry, I'll start again. Yeah, so Mouseview.js is a mouse-based attentional tracking toolbox. And it's made in collaboration with Edwin Dallmeyer, who's at the CVU, and Tom Armstrong, who's in the States at Whitman College. And we kind of made this because recently there's been a move to running eye tracking online. But there's restrictions around that. And we came up with the idea that mouse tracking might be a viable alternative to how all of those things work. Cool. So first is, why do these types of experiments online? So collecting eye tracking data to start with in person is fairly time intensive. Just getting people in and recalibrating and meeting them in person restricts your sample size. And more recently, it's been less possible due to COVID-19 shutting labs down intermittently in different parts of the world. And regardless of those two things, running research into the lab gives you a lack of access to some types of large samples and populations that you might otherwise not be able to reach. So can't you just use a webcam to run eye tracking studies online? Well, you can. There's some very good toolboxes for this. So webgazer.js is one of them. It works by using some image detection techniques to work out where the participants' eyes are and where their pupils are. And then it predicts based on people's mouse movements by default, although you can train it to do a calibration scheme. And then it captures across the trial where it thinks people are looking. Sorry, I should have mentioned before, I'm also a part-time developer at Gorilla. So we implemented this in Gorilla. It's also in JS-Psych, or people have used it in JS-Psych, and PsychoPy have integrated this toolbox as well. There's also other options. So Lab Advanced has their own version of this, which I personally found to be a bit better than webgazer.js. However, it has extremely long setup times. Like you're talking like 5 minutes or so of staring at calibration points before you can even start collecting data, and then you keep having to recalibrate. So all of this means that there's non-trivial issues with collecting data in this way. So there's lots of boring calibration and recalibration, which I found has led or can lead to high attrition from your standard kind of online recruitment platforms. Participants tend to not stay still. So even those that don't leave the study, you lose a proportion of data from them. And then when you get to analyze the data, generally the quality of the data for most participants, so 80 to 90%, is usually only good enough to kind of render heat maps, an area of interest kind of analyses, rather than consistently extract fixation points or kind of time-based information within the trial. So all of this led Edwin and Tom and myself to decide that maybe doing mouse tracking is a better alternative, especially if you're doing top-down attentional studies. And so this is what it looks like. So you occlude the screen, and then you have this kind of unoccluded circle that tracks your mouse. And this forces participants to move their mouse to see the image. And that's a way of kind of restricting their attention and then tracking it to create kind of heat maps or fixation points. Okay, and this toolbox is called Mouseview.js. It's an open source JavaScript tool which enables this occluded mouse tracking. It has two main functions. The first is to generate that overlay that I just showed you with an aperture in it. And the second is to sample and store the mouse positions over time and we have some utility functions for that. It's designed to work on any HTML document, so you can use it on a website for user interface research, or more typically for the people who are listening today, it's probably going to be more relevant in a cognitive psychology behavioral research context. And you can even just overlay it on images in a web page, so nothing complex to get attentional maps. So I've got an example here of just how easy it is to use. So here's the BBC News website, and just by inserting a snippet of code, you wait a few seconds for it to get a screenshot. There's lots of images on here, so that restricts things. And yeah, you can overlay it over this website, and you can scroll around, wait for it to recapture the image and re-render it, and you can even interact with things and click on stuff. Easy to get working. You just need a couple of lines of code. And it's also designed to easily fit into existing experiment builders like Gorilla and JS Psych, which we use a lot here at the unit. So I'll just go through some of the optional settings that you can use. So the first set of settings are to do with the overlay. So you can change the transparency of the background. So you can have the image like semi-transparent, or you can completely block it out. You can set the color, so you can have like a pink transparency. And you can also do a Gaussian blur, which is what I showed you in that example, where it takes a screenshot of everything that's underneath in the browser, blurs it, and then reapplies it. This is experimental because we have to like basically go through each element on the web page and take a screenshot and save that and blur it and then re-render it on the screen. But we provide a callback function in our script so that you can block out the website while it does all of this, and the block isn't captured in that screenshot. You can also change settings for the aperture, so we can allow you to specify that in size in pixels. So you can have a tiny little circle like you can see here, or you can have a really big one. To work between subjects, we allow you to set this as a percentage of page width, so that it will be consistent regardless of screen size, or regardless of the size of the number of pixels someone has on their screen. And we also provide a function to allow you to blur the edge. So it kind of represents these more typical kind of things that you would see in eye tracking studies, where it's trying to replicate the foveated area of vision. We also provide some utility functions. So for data sampling, you could request a sampling rate. Often this will be in increments of 16.66 milliseconds recurring, and that's because that's the standard 60 Hertz refresh rate of most users' devices. And so sampling with more accuracy than that wouldn't make any sense, because that's not actually representing what's happening on the screen. The storage is dealt with by these functions here. So you have a start tracking function and a stop tracking function, and a function for logging events, so things like trial onsets and offsets, and also some functions for session persistence. And so this is if you're going through multiple pages on a website, you can store the data in the browser cache and then retrieve it at a later point. And when it retrieves that information, it automatically adds in some event logging to say this data was from page 1 and this data was from page 2. The most important thing is how do you set it up? So in Gorilla, at the moment we have a code-based implementation, but early next year, January, February, we'll have kind of user interface drag and drop zone. So you can just build it into experiments that you already have or create a new one. We've made some examples that work in JS Psych, and this is using a plugin system. So this isn't like an official JS Psych thing. This is just our implementation of our thing working in JS Psych. So we use a plugin to start the data collection and then a plugin to stop it. And you sandwich your trials in between these. And then you use that event logging function to log in onsets and offsets of stuff during trials. And you can also install it in anything else yourself. So we have a content delivery network for the JavaScript file. So it means you can just copy and paste this into a website or whatever. And then all of those functions I just explained are immediately available, which is pretty cool. And we, so Tom and Edwin and myself have been trying to kind of replicate some eye tracking research just to show how web-based mouse tracking converts eye tracking research in the lab. So we have a paper that was published or that is going to be published soon in Emotion. And we took an experiment from there, simplified it, and just used 2 trials. So this is a disgust avoidance study. So this is consisting of trials where you pair an aversive, disgusting image, like a picture of poop, with a neutral image, and then participants kind of look around the screen where they're looking. And they're also paired with positive images to see how their eye kind of attention changes between those 2 conditions. And this is what the eye tracking study found. They found that over repeated presentations, people would learn to avoid the disgusting images. And that did not happen with the pleasant images. They stayed and they approached them with their eye movements. So the question is did this replicate using Mouseview? So we set up our study in Gorilla. If I click this, I should be able to show you what it looked like. So you had some instructions here. People had to click on, so they started in the middle. And then these pairs came up here. So these are just a neutral pairing to show an example for 10 seconds. So the participants could explore these images, and then it went on to the next trials. So we had 5 discuss pairings and 5 pleasant pairings. Each of them were repeated 4 times. And the idea was we wanted to replicate that effect that we'd seen before. This is kind of what the raw data looks like. So you have all of these like individual samples. And then here, I didn't show a disgusting image because I didn't want to upset everyone with pictures of poop. But here we use the positive image, and you can see on this individual trial, this participant really prefers looking at the positive image compared to the neutral one. And this is the data we got. So we're still kind of like analyzing it. So I haven't broken it down by the number of times that it was repeated, but you can see quite clearly here that in the disgust condition versus the neutral stimuli, people tended to spend less time as you went on looking and exploring that. And that pattern did not occur for the pleasant images. In fact, people consistently looked more at the pleasant images than the neutral images. That was with 150 participants. This is just explaining what the axes were. And also quite positively, we also got them to rate how disgusted they were by those images, and their rating of disgust correlated with the amount of dwell time they had using their mouse. This was a pre-registered study, and this correlation was significant. However, it was of slightly less magnitude than eye tracking data, and we're trying to kind of like explore what's going on there and exactly how that will relate with other conditions. Yeah, so that's it. To summarize, Mouseview.js is a simple tool for attentional tracking. It has examples working in Gorilla and JS Psych at the moment, with a better version coming in Gorilla. And we've preliminary replicated an existing eye tracking study using this. And it's available on GitHub. And feel free to email me if you want to use it. It's still in beta, so I'd prefer it if you were actually going to use it to get in contact so I can help you. Yeah, so look forward to any questions.